I would like to introduce our first speaker, Robert from CryoCloud. Obviously, the title is there. I won't read it out for you, but it's my great pleasure to introduce Robert, who will be talking and taking questions at the end. So thank you very much.
Thank you for the introduction, and thanks to the organizers for letting me speak today. I'm really excited to tell you how we are working towards automating cryo-EM data analysis at CryoCloud. But before I start with that, a little bit about CryoCloud. We have been developing cloud-native cryo-EM software since 2021. Our offering is an integrated end-to-end solution for both data storage and analysis, which we call CryoCloud. And our goal is to automate cryo-EM data analysis to make it more accessible and to provide everybody with the easiest and fastest way to get to the highest quality structures. And we use 3 core technologies towards this goal of automation. That's scalable cloud infrastructure, an intuitive app with automation features, and optimized image analysis algorithms. And we integrate all of these technologies into one platform. This is how CryoCloud works. CryoCloud is a provides fully managed code-free access to dozens of GPUs and petabytes of storage if needed. Once you have an account with CryoCloud, you can simply log in at app.cryocloud.io. You get access to the dashboard where you can upload your data, or you can also use the command line and the terminal. And with the dashboard, you can analyze and manage your data via an intuitive user interface on dozens of GPUs. Importantly, it's code and maintenance free, and you can get started in minutes, and you can also integrate it with your existing infrastructure. But let me show you how it works. So this is a screencast. That's the login page where I'm logging in with my demo accounts. You can have multiple teams at CryoCloud. This is the project table where you can sort, filter, and search all your projects by various parameters, such as storage size. Once you open a project, you see the project grids. We see that here there are 2,700 micrographs that yielded a 3.3 angstrom structures. Now I'm opening the CryoCloud Picker results page. So now the app is requesting the data from the cloud backend. It's not local, and we really invested a lot in making loading times very fast. You can see it's loading the thumbnails at the top and all of the data points. You can view the noise version of the micrograph, adjust the contrast on the fly. You can filter out bad micrographs based on maximum resolution or any other metric, and the number of micrographs will update live. And you can also adjust the threshold for your picking. Now I'm opening the results page of a refined 3D job where we got the 3.3 angstrom structure of CB1, which you can see here. And these are just some features. We have many more, including collaborative features where you can add your colleagues or you can add external collaborators. We have 3D visualization tools for atomic models. And also we have very cost-efficient long-term data storage. This is a benchmark that we published in January 23. Initially, you can see the blue bars. We determined the structure of the GLP-1 receptor to 3.42 angstroms in 3 hours, 17 minutes. We rerun the benchmark several times since. Last time was in August 24 with Reline 5 with Blush. And what we also did is we optimized all the jobs. And when we reran it on the same infrastructure, so same number of GPUs, we could cut down the time in half and got a 3.2 angstrom structure. That's also something important with CryoCloud. You already get an optimized system. You don't need to specify the GPUs, CPUs, or MPIs. We have done that for you, and we are continuously improving it to provide you with the fastest and cheapest data analysis. But this is just one benchmark. Last year, we added workflows as a tool towards automation. A workflow is a sequence of jobs that you can see on the left. This workflow starts at motion correction and ends in 2 refined 3D jobs. And if you look at this part here, you can see which parts we have automated. And everything up until initial model is automated. We use Reliance 2D class ranker for 2D class selection. Only at 3D step, the user has to then select the 3D maps. We're also working on automating that. And the last step is also automated. We then applied this workflow to 9 data sets. For 8 of those, we got structures of 4 angstrom or better with an average resolution of 3.4 angstroms and an average runtime of 7 hours, average cost per structure of $50. Now, many of these complexes are relatively large and have high symmetry, but we did also include 2 membrane complexes, which you can see here on the top left. And the CB1 notably does not have any symmetry. And we got a 3.4 angstrom structure without any manual optimization and also without post-processing such as polishing or refinement. When we did that, we got to 3.2 angstroms. So now we can make the claim that we have solved both the infrastructure issue and can provide an easy and intuitive app with automation feature as CryoCloud is today, so that you can run dozens of data sets in a parallelized and easy way. However, we can only provide this really nice results for easy data. We've been seeing in the last 2 years that most data sets are not the really nice refined data sets that you can find on Empire. So since last year, we have started to work on new algorithms to also tackle those challenging cases. And that's what I'm going to use the rest of the time today. I'm going to speak about the CryoCloud picker, as well as a new anisotropy analysis algorithm that we developed at CryoCloud. The goal with developing our own picker was to have a fully automated and accurate particle picker. So current ML-based pickers are good, but they often require tuning or retraining. And that breaks automation, and it also makes it difficult for new users who are unfamiliar with parameter optimization and also what to expect. So we wanted to develop a picker that is very precise and also very robust / generalizable across many different data sets. On the right, you can see a micrograph that was picked with our CryoCloud picker. Oh, and I would also like to acknowledge Anna, our ML engineer who led this project. On the right, you can see a micrograph picked with our CryoCloud picker default settings, and on the left with Topaz default settings. And you can see that there are much fewer double picks, and it looks much cleaner. But we can also quantify that. So Anna established ground truth for 6 data sets that she manually labeled. You can see in gray here is the ground truth, and in blue the true positives. And while Topaz does a great job at retrieving the true positives at the default settings, we also get a lot of false positives. If we look at the ratio for each true positive particle, we have about 3 false positives. Now, if we look at the CryoCloud picker, it also does an excellent job at retrieving the true positive particles in blue. However, there are way fewer false positives. And if we look at the ratio of false positives to true positive particles, now it's inversed, and we have more true positive particles than false positives. Next, we also quantified the centering precision, since we do have the ground truth, and on X, you can see the mean distance to the ground truth center of our picker and of Topaz. And you can see that there's a fairly large spread for Topaz from about 1.5 to 8 pixels, whereas our CryoCloud picker does much better at finding the true center with a mean distance to ground truth of 1 to 3 pixels. If you look on the y-axis, you can also see the confidence of the model, and it shows that our picker is more confident in the picks as well, which can be important if the noise is higher or your complex is smaller. And these features make it very valuable for dense clusters or small particles. We have also benchmarked this. Here is a data set of the SARS-CoV-2 RNA-dependent RNA polymerase. And it's about 10,000 movies. We did the picking both with Topaz and with our picker. The initial number of particles was fairly similar, 4.3 million for Topaz, 3.4 million for our picker. However, when we did the 2D classification, the difference was very significant. For Topaz, we only see 3 nice classes that have this characteristic RNA stock here, whereas the 2D classes picked with the CryoCloud picker show much more details, and we get many more classes. Out of the nice classes, we get 200,000 and 850,000 particles, respectively. And when we then run the initial model job on those nice 2D classes, we do not get a map with defined features from the topaz picked particles, but we do get a nice map with this characteristic RNA stalk from the particles picked with our picker. Also for this data set, we noticed that there is a dimer class for the topaz picked particles and not for ours, so we contribute that again to our improved centering precision. Then we did another benchmark, and here we said we just pick the very best particles of each model. We don't run 2D classification, no 3D classification. We just pick 1.2 million particles. And from this data set, the publication had about 2 million initial picks. So this was a relatively conservative number. We run initial model job, pick the nicest class, and then run a 3D refinements. From the topaz picked particles, we got a 4.6 angstrom map. Oh, and I should say it's the CB1 receptor, this data set. From the topaz picked particles, we got a 4.6 angstrom map and 330,000 particles. From the CryoCloud picker, we got a 4.2 angstrom map and also 150,000 more particles. If we look at the map, you can see we have much more details. You can see the pitches and the transmembrane helices picked with our picker. And you can also see, if you look at the angular distribution of particles, we have way less preferred orientation. So our conclusion is that this picker is less prone to a bias in the orientation of the particles. So in conclusion, a small summary: our picker is more precise, shows robust picking with fewer false positives. We have better centering precision, and it's less prone to orientation bias. And what we will be adding very soon, by the end of next month, is a retraining feature, because we have many users who really challenge us with difficult data sets, and there sometimes the threshold really needs to be adjusted. So the retraining feature will really help with that a lot, where we then can retrain across many different data sets for specific targets. Motivated by this issue of preferred particle orientation, we then started another project to develop a new measure for map anisotropy. And this project was led by Guido, our director of structural biology. And there are several good tools out there to quantify preferred orientation and anisotropy. There's the sampling compensation factor on the left here, developed by the Liumkis lab, which takes in the angles of your particle sets. We have implemented that in CryoCloud as well. And there's also the conical FSC curve that plots the FSC across different viewing directions. And we have those tools now, but we quickly realized that they are somewhat limited. The measure on the left requires the particle angles, and often users send us their maps and we don't have those, or also if you look at EMDB, you don't have them. And the measure on the right is multidimensional. You cannot simply compare several CFSC curves easily to each other to find any issues. So we decided we wanted a simple 1D measure for map anisotropy. And Guido proposes 3D FSC cylindricity as a metric, which takes the half maps, computes the 3D Fourier shell correlation, cuts off at the gold standard criterion, and then you get these shapes in the 3D FSC space. And you can see that for the EMDB entry on the left, we have a pancake structure, so we have high resolution in X, whereas for the example on the very right, it's a perfect sphere, so we have equal high resolution in all directions. And then the challenge was to develop a metric for that. You can see that with our 3D FSC cylindricity score, it nicely correlates with the observed shape. If we now look at the related EMDB entry, you can see that for this SARS-CoV-2 spike protein on the left, nominal 3.7 angstrom resolution, but I think you would expect a better map at this resolution. You can see that it's highly anisotropic, and you don't see nice side chain densities that you would expect at this resolution. The second example at 3.1 angstrom, it's also a spike protein. It's already a bit better, but I hope you can appreciate that the two examples here on the very right show much more details. You see beautiful side chains. And what's also interesting, if you look at the resolution distribution from 2.9 to 3.1 angstrom, it's very close by, but I hope you agree that the difference in map quality is vastly different. And now we have a new metric with the 3D FSC cylindricity for quality assurance and estimation of the map. And that really correlates nicely also with the observed quality of the map. Next, what we did is that we ran our anisotropy analysis algorithm on the entire EMDB. We set a cutoff at 7 angstrom resolution, and so we analyzed 14,000 maps. And we used our cloud platform to do this analysis in a single day compared to 2 months it would have taken on a single workstation. And you can see the result of this on the right. On X, you have the resolution of the EMDB entry. And on Y, you have the 3D FSC cylindricity score. And you see very nicely that for very high resolution entries, the 3D FSC cylindricity is very low. But once starting from 2.5 to 3 angstrom and lower resolution, anisotropy increases. And that's in line with what we would expect, because likely what happened here is you have preferred orientations that limits the map resolution and induce anisotropy. Next, we asked ourselves, can we identify and improve maps with high anisotropy? And we found this entry. It's a group 2 intron retrotransposon map at 3.1 angstrom, published by Chung et al. In Science from 22. And it showed the 13th highest anisotropy across the entire EMDB. And despite its relatively high resolution of 3.1 angstrom, it had a very low Q score of 0.262, which is already a bit suspicious. The analysis was previously done with Topaz and CryoSpark's non-uniform refinement. So we decided this is a great case to test our own CryoCloud picker and reline with Blush to see if we can improve the map quality. And I would really like to credit and thank the authors for uploading this raw data. This is not about bashing the previous map, but it's about learning, especially from this challenging sample. So thanks to them. You can see the map that we obtained on the right compared to the previous map. So this is a DNA complex. Now, I hope you agree with me that we can see much more detail. We can resolve the nucleobases now, which are blurred here. If we look at the cylindricity score, it's also lower, and the CFSC curve looks much better. So overall, a much improved map. However, if you were to look just at the resolution, you would be easily misled, because here we measure 3.4 angstrom versus 3.1 here. If we zoom in more into the map, again, you can see that we have the blurring here of the nucleobases. Here they are nicely resolved. And also we can see density for this base pairing here that we don't see here at the given resolution. And we have seen really a lot of impacts using Reliance Blush for many maps that show anisotropy. So also big shout outs to the authors there. In summary, CryoCloud is a complete end-to-end cryo-EM data solution. We provide improved algorithms for cryo-EM data analysis, such as our CryoCloud picker and our anisotropy analysis, with which we did the first systematic anisotropy analysis of the entire EMDB to our knowledge. I did not speak much about security, but we also provide enterprise-grade security and recently got the ISO certificate. If you want to try some of our algorithms, we have a free plan for academia, which comes with 1 terabyte storage and 5 compute hours. And the jobs, the picking and the anisotropy jobs, they're very fast. They run in minutes. So you can really analyze a lot of your data using that. With that, I'd like to thank you. Please come to our booth. We also have a raffle where you can win a 3D printed protein structure of your choice if you scan the QR code and fill in this form. And lastly, I would also really like to thank our great team. What makes us strong at CryoCloud is that we combine expertise from many different domains, including structural biology, software engineering and machine learning, and computer science. Thank you.
Thank you very much, Robert. Are there any questions? Okay. Somebody else wants to do, sorry, myself. I'm going to cheat quickly and ask my own question first. So the automation, obviously, I'm from industry, so automation is always going to really excite me. What more do you think is, I mean, what's the natural next step for the automation as you see it?
Yeah, so that's what we're working on right now, working on analyzing more data sets just to get more data points and new algorithms as well. So the idea is that we want to get to the same resolution that has been previously published, and how we are approaching it now is to look at specific targets. Yeah, if you want to know more, hopefully by the end of the year, we have something exciting coming. You have to invite me again to speak again. So
okay, that works for me. Brilliant. Thank you.
In the 2D and 3D classes, some of the classes that do not look good, but probably some of the particles are still fine, some of them are not good. Is it any possible to separate them so that we can recover more good particles?
You're referring to the SARS-CoV-2 RNA polymerase, the 2D classes, or?
Any particles, any kind.
So what we do see is that if you go in with a clean particle stack, the 2D classification significantly improves. So that's what we focused on right now, to really just get as clean of a particle stack as possible. And that will translate into all downstream steps. Yeah, if you start with bad particles, you have this issue that you pull some good particles into bad classes and the other way around. That's something we need to explore more. Yeah.
Thank you. And I have another question. Is it possible to combine this application with other like Reliant, like Crosspark, so that we can process some data in other applications and do something here and do something in another app again?
Yeah, definitely. So we have users that do hybrid use. New data sets come into CryoCloud. They can also download all results. We don't obscure the data. So if you want to download your picking coordinates and then continue analysis locally, that totally works. Yeah, we support that.
Thank you.
Are there any other questions? Oh, sorry, right back there.
Hello, at the back over here.
Back.
Hi.
Hi.
Very nice talk. The anisotropy measurements are nice. Are you writing those up and publishing that so others can benefit from that assessment?
Yeah, we aim to publish that in Q3, hopefully, if things go well. Yeah.
Looking forward to it.
Final question there, please.
Maybe it's too early. Are you considering expanding to the ET as well, ET processing?
We do have a prototype for cryo-ET. So yeah, please reach out if you want to know more there.
In which case, thank you very much, Robert.